Welcome back friends. Katika kipindi kilichopita tulikuwa tunaangalia kuhusu evidence for the location of the hereditary material in the chromosomes. Na kipindi hicho naenda kuendelea na properties of hereditary materials. Kwa hiyo properties of hereditary materials ziko kama 6 hivi. Na kwenye kitabu cha Tai wameziandika tu bila kuzielezea, lakini kwenye hizi notes zina maelezo. So it was proposed that proteins and nucleic acid could be hereditary materials that carry information from parents and handle them to offspring. Lakini pia hata katika evidence iliyopita tulisoma kwamba the chromosome analysis showed that chromosomes contained protein and DNA, which is nucleic acid. But it was unknown kwamba which one among the two is the genetic material. It's whether DNA or it is protein. Unaona. So later proteins were excluded from being hereditary material since they lack properties of genetic material. Proteins, they were excluded simply because zilikuwa hazina properties za genetic material. Wakati DNA ilikuwa inaonyesha na properties za genetic material, protein ilikuwa haionyeshi hizo property. Now what are the properties of genetic material now, or the characteristics of hereditary material? First is the storage or carriage of information. The storage or carriage of information. Hereditary material kazi yake ni kuchukua information from parents to offspring. That is the major rule of hereditary materials. Na ndio maana unaona hapo imeandikwa storage au carriage of information. Kwenye kitabu cha Tai utakuta wameandika they contain hereditary information in the coded form in their specific structure called genes. They contain hereditary information, that means they carry hereditary information. But if you are asking a question of explanation, kujibu kwa kutumia maelezo haya itakuwa ni better rather than summarizing how Tai wameandika. The hereditary materials, they contain information in the coded form in their specific structure called genes for the control of biological functions of the cell. Kama ambavyo nilisema kwenye topic ya cytology form 5 kwamba the nucleus control all activities of the cell because of the presence of DNA. Always DNA tend to express or tend to control other activities of the cell by the way of producing RNA, and this RNA always tend to form protein. Unaona, kwa DNA inatengeneza RNA. RNA inaenda kubadilishwa kwenda kuwa nani? Protein. Hiyo tunaita ni central dogma. Hiyo tunaita ni central dogma of biology au central dogma of molecular biology. From DNA to RNA then to protein. DNA inatengeneza RNA, RNA inabadilishwa kwenda protein, and that protein is the one which is responsible to regulate other activities within the cell. That is the central dogma of biology or central dogma of molecular biology. Unaona? So the central dogma of molecular biology believes that DNA can be copied into the complementary strand, which is RNA, and this RNA can be translated into protein. This protein now controls different activities of the cell. That is the central dogma. So hereditary materials store and also express its information. Kuna vitu viwili hapo, kuna kustore pamoja na kuexpress. The storage of information is done by DNA, but the expression of information is done by RNA. So RNA express easily the characters in a form of protein. So DNA requires RNA for formation of protein. DNA being more stable is considered better than RNA for the storage of genetic information. However, for transmission of genetic characters, RNA give better results. So DNA actually it store genetic information, but RNA it transmit genetic information or it translates genetic information. Manake kile kilichochukuliwa kwenye DNA lazima ikitengeneze copy yake kio kwenye RNA. RNA ndipo itengeneze protein. Kwa sababu mwisho wa siku ni kwamba Unapokuja kuona nywele zilivyokaa kipilipili ni nywele gani, hair demarcation, that is because of the protein keratin in the hair. Unapokuja kuona labda the color of skin, it's whether albino au kawaida, it's because of the presence of melanin pigment in the skin. What is melanin? Melanin is the protein. So almost everything in the body we are talking about, or almost every character of human being or even other organism, we are talking of the proteins which result from the RNA. And this RNA does not produce protein by itself. It takes instruction from the DNA. So DNA tend to store hereditary information, but from the storage in the DNA, what happens is that the information stored in the DNA, they are copied to the RNA, and then RNA transmits this genetic information and expresses them in terms of protein. The second property of hereditary material is self-replication. Self-replication. Now, ukiangalia kwenye kitabu cha Tai, hiyo ni point number 3 wao kwenye kitabu cha Tai. Wanasema they have the ability to replicate or form their own copies. That is self-replication. Self manake ni kitu ambacho kinafanya chenyewe. Self kinafanya chenyewe bila kuwa control na kitu kingine. So that is self-replication. In the presence of appropriate enzymes, the hereditary materials are capable of undergoing self-replication without losing any of information. Hereditary materials are capable of replicating exactly so that the genetic determinants are transmitted down from cell to cell and from generation to generation unchanged. Other molecules in the living system such as proteins fail to exhibit this property. Unaona? Kwa hiyo molecules like protein, they fail to exhibit this property. That's why protein ilitolewa kwamba that's not hereditary material. Why? Kwa sababu ilikuwa haionyeshi uwezo wa kureplicate yenyewe. Now remember, again nikwambia, we have two categories of nucleic acid which are hereditary material, the DNA and the RNA. But the major hereditary material we are talking about, it is the DNA. And it is the DNA which is capable of replicating itself, forming its own copy. And as we shall study later, ni kwamba during the process of DNA replication, sometimes the errors they can occur. Unaona, in the place of thiamine, it can be replaced with another amino acid, another base. Unaona, kwa errors katika DNA replication huwa zinatokea. Lakini there are different processes to correct these errors, and there are different mechanisms to correct the errors which result from DNA replication. Hizo mechanisms kwa level yetu hatuzisomi, lakini ukifika chuoni utasoma the mechanisms which are used by the body to correct the errors which happen during DNA replication. So DNA is the genetic material which is capable of replicating itself. Actually, the RNA cannot replicate itself until it is formed from DNA. So when you are talking of self-replication here, we are talking more of DNA, which is our major hereditary material. This is capable of replicating itself and forming its own copy, exact copy. Copy sawa na original. Na nimekwambia kwamba hii copy ikiwa na makosa, basi kuna mechanism ambayo ya makosa yanatengenezwa ili kuweza kutengeneza the exact copy. The third characteristics of hereditary material is ability to undergo mutation. Ability to undergo mutation. Ability to undergo mutation. Kwenye kitabu cha Tai, hii ni point number 6, imeandikwa they undergo occasional changes or mutations in the structure and the functioning of their genes which are of permanent nature and inheritable. Unaona, wameandika hivyo, hapo wameandika pale mbele kwamba this tendency is important for evolution and adaptability of the organism. So tumeandika ability to undergo mutation. So mutagens, tukisema mutagen, a mutagen ni kitu kinachoweza kusababisha mutation. Mutagens can change the structure of genetic material and DNA. Mutagens are agents which cause mutations in living organisms such as X-ray, gamma rays, and etc. Also, mutation can be due to replication error. Yani katika kureplicate nimekwambia DNA inavyofanya self replication kuna errors zinaweza katokea. Hasa some errors they can be corrected, lakini some errors they can't be corrected. And if these errors they are not corrected, that means mutation occurs. So mutation, it can be due to replication error au inaweza kuwa due to mutagens. Genetic material have ability to undergo mutation, hence giving more variations which are important for evolution. However, RNA mutates at faster rate when compared with the DNA. RNA inafanya mutation faster ukilinganisha na DNA. So viruses with RNA genome therefore show mutation and evolution at faster rate and thus have shorter lifespan. This is the reason why it's very hard to treat HIV virus. HIV virus, it is the retrovirus. It is the retrovirus. Unaona? Na ndio maana hata zile dawa za kupunguza makali ya virusi vya ukimwi zinaitwa ARV. ARV ni anti-retrovirus. Anti-retrovirus. A ni anti, R ile ni retro, afu V ni virus. Anti-retrovirus. Retrovirus ni virus gani? Retrovirus ni virus wenye RNA genome. Kwa sababu virus wako aina mbili. Wako wenye DNA genome, wako wenye RNA genome. Hao wengine virus wengine acha nisiongelee kwa level yako. Kwa hiyo jua tu kwamba utoka ulivyosoma kwenye classification, virus they can either have DNA or RNA. Retrovirus it has RNA genome. Lakini kwa mfano retrovirus akiingia kwa human being na anavyotaka kureplicate, lazima ile RNA yake ibadilishwe kwenda kwenye DNA, na ndipo ile DNA iliyotokana na RNA ya virus, ile DNA incorporate au iungane na DNA ya human being, na baada ya kuungana nayo ndio baada ya hapo process nyingine zinatokea, na hapo kuna enzyme huwa inaitwa reverse transcriptase enzyme. Reverse transcriptase anafanya reverse transcription from RNA to DNA. Transcription ni from DNA to RNA. Yeye anafanya reverse transcription ya kutoka RNA to DNA. Lakini the reason behind why it's very hard to treat ARV is because ARV is, why it's very hard to treat retrovirus or HIV virus, it's because HIV virus is RNA virus, it is retrovirus. Unaona, retrovirus ni kundi kubwa la virusi wanaotumia RNA kama genetic material. So since HIV virus is the retrovirus, it mutates faster, inafanya mutation faster. So when they make a medication for one category of this retrovirus, kwa sababu even HIV virus they are virus categories. There is HIV A, HIV B, HIV C, HIV D, AC, and other mutations, and several mutations they result there. So when they try to make a medication for one variant of the virus, after a few months a new variant inatokea. Na hasa inapotokea ni kwamba unakuta mtu mmoja let's say ni mwanaume ana variant fulani ya HIV, let's say HIV A, hafu mwanamke ana variant nyingine HIV, let's say HIV C. Unaona, hapo watu wanazini. Wakishazini ya watu wanatengeneza labda mtoto ambaye anakuwa na HIV mutant mwingine. Unaona, yule mtoto akiwa affected anaweza akawa ana mutant mwingine wa HIV, sio tena A wala sio C. Unaona, can be AC or can be a new category of mutation. So the new categories of retrovirus, it is because people they are interacting. People are interacting. Squeeze wazungu wa Afrika wanazini, wanaoana, and some of them they are affected with the virus. So that's why this lead to the generating new categories of the retroviruses. So mutation are important for evolution and adaptability of the organism to the environment. In case organism yuko ana live kwenye environment ambazo they are not such much suitable for the organism to live for a long time, mutation they are very important for the adaptability of an organism. However, sometimes mutation ndizo zinazofanya pia retrovirus awe ni vigumu kutibika. So in such cases mutation they are non-advantageous. Number 4 is the high degree of chemical and physical or structural stability. When we are talking of the physical and the structural is one thing. So tunazungumzia kwamba the hereditary material, they have high degree of chemical and physical au structural stability. Katika kitabu cha Tai hiyo ni pointi number 5. They are stable both chemically and physically. Now in this case tunazungumzia sasa, nimekwambia ni tunazungumzia sasa DNA. However, RNA is also a genetic material, but the one which is more chemically and physically more stable, it is the DNA. And let's start explaining here. You understand why DNA is used for the storage of hereditary material, or for the permanent storage of hereditary material, while RNA is used for the temporary storage of hereditary material. So any material which is responsible for transferring information from one generation to another must be extremely stable. It must be extremely stable. If it were altered to any extent, then imperfect copies would be made. Manake kama haitakuwa stable, manake itatengeneza copy ambazo ni imperfect, copy ambazo hazijakamilika. So unlike proteins, DNA show remarkable metabolic stability. Proteins zenyewe haziwezi kaonyesha stability kama walionayo DNA. So DNA they show a remarkable metabolic stability. Example, ATP molecule can be broken down to ADP and finally to AMP. So ATP is not genetic material, although it is a nucleic acid. Yani ATP ni nucleic acid. Unaona? Component za ATP zinafanana na component za nani? Za DNA. But ATP, it has the same component as the DNA, but it is not the genetic material. Why? It is because it can easily be broken to other component. So simply because ATP can be easily broken to other component, then it is not qualified to be the genetic material. Even in Griffith experiment, tutaisoma hiyo Griffith experiment of transformation principle. Itaenda kuisoma mbele kidogo. The Griffith experiment of transforming principle. DNA survived in heat that killed bacteria. Yani unachukua bacteria unawahiti, lakini bacteria anakufa, DNA inakuwa bado iko stable. Why? It's because unaona such heat at least did not destroy some of the properties of genetic material. That means it is the heat which can kill bacteria, but the DNA within the bacteria can still be stable. And if this DNA is transferred to another bacteria which is living, DNA inaendelea kufanya kazi kama kawaida. Unaona? Kwa hiyo DNA is very stable. This now can easily be explained in light of DNA that the two strands being complementary, if separated by heating, come together when appropriate conditions are provided. Yani hata ile DNA ukaihit ukaseparate zile strand, unaona strand mbili kama ambavyo tutaenda kusoma kwenye structure of DNA, ukiseparate zile strand mbili za DNA, endapo tu condition zikarudi kawaida, zile strand zinajikoil tena vile vile. However, the second hydroxyl group present at every nucleotide in RNA is a reactive group. Yani RNA ina sugar ambayo ni ribose, while DNA ina sugar ambayo ni deoxyribose. Tutaenda kusoma katika chemistry of hereditary material or the chemical composition of hereditary material, tutaona hivyo vitu. Unaona? So the hydroxyl group which is present in carbon 2 of the ribose sugar, it is the reactive group and makes RNA liable and easily degradable. Unaona? RNA inakuwa ni liable na ni rahisi kuivunja RNA kuliko DNA. So RNA is also now known to be catalytic, hence reactive. Unaona? RNA it is catalytic kwa sababu gani? Yenyewe ni reactive, ina react kirahisi. Therefore, DNA is chemically less reactive and structurally more stable when compared to RNA. That's why tunasema DNA is used for the storage of genetic material, Why? It's because it's more stable, where RNA is used for the transfer of genetic material. In other words, DNA is used for the permanent storage of genetic material, where RNA is used for only temporary transfer of genetic material. Therefore, among the two nucleic acids, the DNA is better genetic material. DNA is a better genetic material. In fact, the presence of thiamine at the place of uracil also confers additional stability to DNA. Thiamine at place of uracil in a confer additional stability to DNA. For the same reason, DNA is used for storage of genetic information, while RNA is used for transfer of genetic material from the nucleus to the cytoplasm. Actually, this is not the transfer of genetic material, but it is the transfer of genetic what? Information. So DNA is used for storage of genetic information, while RNA is used for transfer of genetic information from the nucleus to the cytoplasm. Now we can continue with the property number 5, which is the constancy within a cell. Constancy within a cell. Kwenye kitabu cha taa hiyo ni property namba 2. Wanasema the structural element of genetic material are ubiquitous in their distribution. That means they are for everywhere in life form. Constancy within a cell. Analysis show that the amount of DNA remain constant for all cells within a species except for gametes, which have almost exact half of the usual. They have almost exact half of the usual. I think kuna property ya 6 ambayo nashangaa kwamba huku sioni, lakini I remember kwenye notes zangu ilikuwepo. Wewe niangalia hapo kwanza kwenye tai. Yeah, okay. Sawa, ni hizo 5. Then the same quantity, the quality in all the somatic cells of the healthier individual, ndio constancy within the cell. And this property number 2 and property number 4, they are actually similar, lakini sometimes inaweza nikatafuta more detailed explanation na nikaziweka property 6, lakini these are the properties of genetic material. So thank you, and we are ending up here. In the coming session we discuss about the chemistry of hereditary material or the chemical composition of genetic material. Genetic material zimetengenezwa na nini? Thank you, unless we meet in the coming session.